Welcome to the Deep Dive, your shortcut to being well-informed. Today, we're diving into something massive. How do we feed everyone, especially in the toughest places? And right at the center of it all, smallholder farms. I mean, it's incredible. They make up 80% of farms globally. Tiny plots, usually less than 2 hectares, but they're really vulnerable, especially to things like nutrient deficiency. And today, we're zeroing in on one specific nutrient, phosphorus, P. It's absolutely essential for plants. Can't grow without it. But the problem is it often gets locked up in the soil, unavailable. And this is a huge issue, particularly across Africa in those tropical and subtropical soils. You've got intense weathering, breaking down rocks, high P fixation where the soil just grabs onto it, and then leaching, erosion. Just washes away.
Exactly. And when you layer those environmental challenges onto the economic realities for smallholder farmers,
Yeah.
things like, not being able to afford fertilizers,
Yeah.
you see why these farms in Africa are so incredibly susceptible to P deficiency. It's a direct hit to their food production.
Which affects everything, livelihoods, resilience.
It's fundamental survival, really.
It really is stark.
Yeah.
And it makes finding sustainable solutions so urgent. So that's our mission for this deep dive. We want to explore some, frankly, innovative strategies using microorganisms, tiny helpers to unlock phosphorus for plants. We'll unpack 2 main angles. First, bringing in microbial inoculants, basically adding helpful microbes. And second, boosting the microbes already in the soil, the natural ones, through better farm management. And this is all based on some really fascinating recent field studies from across Africa. Okay, let's get into it, this world beneath our feet. Right, first up, strategy 1. Microbial inoculants, adding the good bugs, as you said. So the idea is you introduce specific strains, microbes that are often found in low P soils anyway, hoping they'll make more phosphorus available. Like a soil probiotic, we've seen examples, right? Things like Bacillus teculensis helping chickpeas in Algeria, or Burkholderia, Pseudomonas in Kenya, actively solubilizing P.
Yeah, and a really common type you hear about is arbuscular mycorrhizal fungi, AMF. These are amazing. They team up with the roots, basically extending them, creating this huge underground network to gather nutrients. It means the plant might need much less P fertilizer. We've seen this help soybeans in Nigeria and Ghana,
Oh, interesting.
and cassava too in Kenya and Tanzania.
Okay, but here's where it gets really interesting, maybe even a bit counterintuitive. For ages, the focus was on microbes dissolving phosphorus directly, right, their superpower. But newer evidence suggests that maybe the bigger contribution comes from something else. Like the simple life cycle of microbes living, dying, releasing nutrients, that's microbial biomass turnover. And also just the sheer reach of those mycorrhizal fungi networks, extending the roots.
Right.
These factors might actually be more important for getting P into the plant than the direct dissolving of inorganic P.
It makes sense when you think about it from the microbe's perspective.
Exactly. Why waste energy dissolving P unless you need it? So maybe we've overestimated the impact of things like organic acids from microbes dissolving rock phosphate in the field. It's a big shift.
It is a fascinating point about the mechanisms, and it ties into the effectiveness question with AMF. It can be, controversial and definitely site-specific. Very much so. Like one study found high AMF colonization in common beans when P was low, which you'd sort of expect. The plant's calling for help.
Makes sense.
But interestingly, adding just a moderate amount of P fertilizer, like 15 kilos per hectare, actually seemed to boost the benefits from the AMF.
Oh, okay. So it's not always an either.
Not necessarily. In other studies, on cassava, for instance, found AMF colonization didn't always drop off even when more P was supplied. It's complex. Really shows how much the specific farm environment matters.
And it's not just the direct P solubilizers or AMF. We also see N fixers playing a role, right? Like rhizobia, used a lot in legumes like soybeans in Uganda, common beans in Madagascar.
That's right.
Their main job is nitrogen, obviously, but they seem to help with P too, maybe indirectly.
Yeah, one way is by encouraging those mycorrhizal fungi to colonize the roots more effectively, sort of prepping the ground for them.
Okay, but this leads to a tricky research problem, doesn't it?
It certainly does, a big methodological challenge. Because these inoculants, they're almost always applied with P fertilizers or with compost, manure, other organic stuff. So trying to tease out exactly what the bacteria alone are doing for P cycling, it's incredibly difficult in a real-world field setting.
So you can't easily isolate the effect.
Precisely. Which hammers home that need for site-specific strategies. The local environment plays such a huge role. How confident can we be about the inoculant's true impact in these mixed systems?
That's a really crucial point, and it makes you wonder about practical adoption. For the smallholder farmer, what does this mean? Even if they show promise in labs or specific trials,
Hmm.
you've got huge barriers in Africa. The market for these things is underdeveloped.
Mm,
The cost can be prohibitive for farmers on tight margins.
absolutely.
And there's still this underlying uncertainty about whether they'll actually work consistently across different farms, different seasons. Will they really reduce the need for expensive fertilizer enough?
That's the key question.
So it really boils down to needing rigorous testing. Field testing in specific contexts to see what actually works on the ground. Okay, so inoculants are one avenue. Promising, but uh, clearly complicated. Which brings us to the second major strategy. What if it's not about adding microbes, but working better with the ones already there? Enhancing the natural soil microbiome through sustainable farming practices. And a huge part of this is organic matter management. Things like compost, manure.
Vital. Absolutely vital for smallholder farms, boosting productivity, resilience.
And sometimes the studies show the effect can be even better than mineral fertilizers alone. Is that generally true or just sometimes?
That's a good question. It's nuanced. When organic matter does outperform, it's often because it's doing more than just supplying P directly. It's stimulating the microbes. It boosts their numbers, their activity, which helps break down organic forms of P already in the soil, making it available. Mineralization. Plus, it can encourage those P-solubilizing bacteria and fungi we talked about. And even at a genetic level, increase the expression of genes involved in P cycling, basically switching on the microbial machinery.
That's fascinating. So it's feeding the microbes, which then feed the plants.
In a way, yes.
And the type matters, right? Manures versus plant material.
Generally, yes. Animal manures tend to have more P and nitrogen compared to plant residues. They can lead to higher microbial activity, more P release, especially if they have a low carbon to phosphorus ratio, less carbon for the microbes to chew through relative to the phosphorus available.
Got it. And there was that example, Tithonia diversifolia, a green manure.
Yes, that's shown some really positive results, boosting microbial turnover and releasing plant-available P. A good local resource in some areas.
But there are challenges here too, I imagine.
Oh, definitely. One issue is just the variability. Compost here is different from manure there, which is different from green manure somewhere else. Their composition varies hugely across studies.
Yeah.
So separating the purely physical or chemical effects on the soil from the purely microbial effects, it's hard.
Right. Is it the structure change or the microbes?
Exactly. And again, strong interactions with local climate and soil type. What works well in one place might not work as well elsewhere. And that fundamental question remains, how much can this microbial P release really offset the need for external P inputs? We're still working that out.
Okay, beyond organic matter, what about planting strategies? Mixed cropping comes up a lot.
Yes, another powerful tool.
Combining different plants, like maize with a legume, say cowpea. The idea is they use P differently, more efficiently together, improving availability both spatially, like roots exploring different soil areas, and biochemically. How do microbes fit in?
Oh, they're deeply involved. It's not just about root architecture. Mixed cropping systems consistently show increased microbial activity and biomass, more life in the soil, which, again, leads to more P release, better P availability overall. These diverse plant systems also tend to foster more diverse and beneficial microbial communities, promoting those plant growth, promoting microbes.
Like creating a better team underground.
Precisely. A more functional team.
And there are some cool examples, right? Like
Yeah.
integrating annuals and perennials, millet and shrubs in the Sahel.
Yes, Guiera senegalensis shrubs with millet. Studies show this enriches the soil with bacteria and fungi known to promote plant growth.
Wow.
And even more complex systems, like agroforestry home gardens, you see really high microbial diversity. Work in Ethiopia found lots more AMF spores, greater diversity in those multi-layered systems.
Which likely helps with pea uptake.
Very likely. It's incredible, isn't it? This whole hidden world working away. Imagine tapping into that more effectively.
Absolutely. Okay, one more practice. Tillage. How we prepare the soil. Conservation tillage, reduce till. No-till gets a lot of attention. Good for protecting soil, saving water, building organic matter, all critical for smallholders.
And there's a clear microbial link. Reduced tillage generally boosts microbial biomass and diversity compared to conventional plowing. We've seen this lead to higher microbial P turnover, better P availability in places like Western Kenya. And no-till systems often show increased activity of important enzymes, like phosphatases, which break down organic P.
Okay, that sounds pretty clear-cut. But is it always? I seem to recall some conflicting findings.
Ah, yes. This is where it gets particularly complex.
Because while intensive tillage can disrupt fungal networks and soil structure, other studies actually show it can sometimes boost the breakdown of organic matter, which might release P, at least in the short term.
That's true. Mineralization can sometimes be stimulated by disturbance.
And some reports even found higher enzyme activity or microbial counts in certain traditional tillage systems like tillaging compared to no-till.
Exactly. So you have these varied, sometimes contradictory effects on the soil biology,
Which means?
which means the yield outcomes aren't always consistent. Tillage definitely shapes the microbial community, huge impact there, but those changes in the microbes don't automatically or reliably translate into better yields for the farmer every single time. It's not a simple A to B relationship.
So it's not as simple as no-till is always best for microbes, therefore yields.
It appears not always, or at least the link isn't always direct or immediate in terms of yield. Soil biology is dynamic.
And practically speaking, adopting strict no-till can be tough for smallholders, right? Needing specific equipment, maybe new skills.
Very true. Access to tools and training can be major hurdles.
So what's the alternative? I read about just tilling less often.
Yes, extending the intervals between tillage. That's been proposed as a more accessible first step, a pragmatic approach towards conservation.
Okay, so wrapping things up here. We've seen phosphorus deficiency is this massive hurdle for African smallholder farmers. Depleted soils, often limited access to fertilizers.
It remains a core challenge. And while these micro-based strategies, both adding them and nurturing the ones already there, show real promise across Africa's diverse farms, we have to be honest about the complexities. Pinpointing that microbial P mobilization is the direct cause of improved plant nutrition. That's still really challenging, scientifically and practically.
So looking ahead, what's needed? More research, obviously.
Definitely. Comprehensive field trials for inoculants are key.
And maybe better tools to see what's actually happening, like metagenomics to map microbial genes.
Yes, understanding the functional potential. And isotopic labeling, actually tracking P atoms as they move through the soil and into the plant. That gives much deeper insight into the mechanisms.
Getting a clearer picture of the pathways.
Exactly. And on the other side, with sustainable practices like organic matter use or conservation tillage, they're incredibly promising for soil health and microbes. But adoption hinges on overcoming real-world barriers. Access to compost or manure, access to the right tools for planting or reduced tillage. Bridging those gaps is absolutely crucial.
It really brings it back to the practical realities on the ground. So here's something to think about. Given what we've learned, the surprises about how microbes really mobilize phosphorus, the complex effects of farming practices we thought we understood, what kind of innovations or maybe collaborations could genuinely bridge that gap between the science and the farmer's reality in Africa? How do we make sure these beneficial microbial processes can actually become transformative for food security on the continent?